This lesson is all about the process of cell division. All right, so all cells store genetic information in chromosomes. Now, here we have a prokaryotic cell, and it's a little different than a eukaryotic cell because its DNA is packaged into a single circular chromosome, and it just kind of floats around in the cytoplasm, as you see here in this illustration. However, with eukaryotic cells, the DNA is actually packaged into multiple chromosomes. And so what we see here is an artist's illustration of the structure of eukaryotic chromosomes, and they kind of represent a butterfly or an X shape. And the amount of organization within that chromosome is really, really phenomenal. There's a ton of data in there, and it contains information that helps make you you, at least part of it. So the organization, let's break it down a little bit. We start with what's called the DNA double helix. And the double helix is something that was discovered actually not that long ago by a couple of gentlemen who I believe both are still even alive today. So this is new information in the sense of, the whole history of humankind. This is really relatively new information that our DNA is packaged in what we call the double helix format. But then we've also got these histones. So basically what happens with the DNA is it gets wrapped up into these coils that you see over in this area, but they get wrapped around these histones. And these histones are important. They're a special type of protein, and it actually helps to prevent the chromosome from getting all tangled up with itself. I don't know if you've ever tried to roll up a ball of string, and then when you unroll that ball of string, it tends to be kind of a mess. Gets all knotted up and everything, or maybe even a necklace or something like that. If you've had experience with that, you kind of know what I'm talking about. Now, the histones, again, the DNA kind of wraps around those histones, and they basically uh, produce what we call a nucleosome. You see that here in this area. Let's not get too far ahead of ourselves. So we got the nucleosomes here. So that's kind of a bunch of histones with a bunch of DNA wrapped around it. So they're actually helping to prevent all of that tangled up. Now, we were talking about taking a ball of string and rolling it up, and when you unroll it, for some reason, it's all tangled up. But if you were to wrap it around a spool, like you get a thread from a spool, right? If you wrapped it around that spool, it would not get all tangled up. And so that's kind of the same idea here with these histones. They help to keep the chromosome organized. Now, once they begin to wrap, they are called coils. So we have these coils. And then the coils actually wrap up as well. So it's like a coil on a coil. And we got these supercoils. That's what we call those. So back here, we talked about nucleosomes. And again, they're made up of DNA that's wrapped around multiple histone molecules. So the nucleosome refers to both the chromosome and the histones. Okay, so it's kind of a combination deal there. So I just wanted to point that out to you. And once we tightly pack those nucleosomes together, that's what we call the coils. Okay, and then as we finish everything off, as the supercoils develop, we end up with these duplicated chromosomes, and each of these ends is called a sister chromatid because they're basically the same information. So they're sisters. and they're connected. The four legs or wings of the butterfly, if you will, they're all connected at what's called the centromere. We'll get into this in a bit more detail as we move through this lesson, but for now, let's just stick with a duplicated chromosome made up of supercoils, which are made up of coils of nucleosomes. And nucleosomes are basically the DNA double helix being wrapped around histones. All right, so let's talk about prokaryotes for a moment. Now, we have a number of steps that prokaryotic cells go through in order to divide, and this process we call binary fission. Binary refers to the fact that it's two, right? Splits one thing into two. Bi means two, right? So binary fission means to separate or to divide, and so this is splitting or dividing a cell into two. And this is what prokaryotes typically go through. This is not, however, how things work in eukaryotic cells. We'll get to those in a minute. Prokaryotic cells, because they're single-celled organisms usually, means that this process can be quite simple compared to, say, eukaryotic cells, which are cells that make up a living organism. Multiple cells make up the living organism. All right, so back to prokaryotes, though. So we start with, we've got the DNA inside of the cell membrane. That DNA will duplicate, and why do you think it is that the cell duplicates its DNA? Does anybody have any thoughts on that? Think about it for a minute. You might want to pause the video and give it some thought, and then hit play again, and we'll pick up where we left off. Okay, so back to our question, why does the cell duplicate its DNA? The answer to that is so that when the cell duplicates or splits in half, each of what we call the daughter cells, the two new cells that are made out of the one, they're each going to have a complete copy of the original cell's DNA. So it has to duplicate it, so each of the new daughter cells has its own copy of the full complement of DNA. So once the DNA duplicates, the two molecules of DNA will attach to different regions of the cell membrane, and then the cell will basically begin to pinch inward and split apart. So what do you think would happen if the cell membrane did not do that indentation piece and then pinch off? Well, the answer to that is pretty simple. It wouldn't form two new cells. It'd still only be one cell. However, the original cell would now have two complete copies of its DNA. Now, again, this is a simple cell cycle, even though the diagram isn't drawn as a cycle. It is a cycle because these two cells here will then go through this same process themselves. So it is a cycle, but we're just seeing one pass through the cycle. But each of these new daughter cells will also go through binary fission. So is binary fission an example of sexual or asexual reproduction? Well, if you're thinking it's asexual, you would be right. There's not two sets of DNA coming together to form a new set. There's not two parents involved. There's only one. And so this is considered asexual fission, asexual reproduction. And also, the thing that indicates that this is asexual reproduction is the fact that both of these daughter cells are 100% identical to each other. There's no difference between the two. And that's a very important distinction. All right, let's move on to the eukaryotic cells. They're a bit more complex. And as you can see, there are a number of phases or steps within the eukaryotic cell cycle. So we're going to go through each of these one by one. Now, most of the lifespan of a cell is spent in the process called interphase. Now, you can see that interphase is represented by this external arrow here, and it only stops when it gets to cell division itself. So interphase is actually made up of 3 other phases. Okay, so these are, I guess you could call them subphases, if you will, but we don't really call them that. We just call them phases. So G1 phase is one of the 3. S phase is the other one. G2 phase is there as well. So the G1 phase is the phase where there's, I was going to say, not a whole lot going on, but that's only in terms of the cell dividing. It's not really doing much in terms of cell division, but it is growing. The cell's getting bigger. Okay. Now, if you think about that, when the cell divides, it's going to be half the size it was before it divided. And that's the point at which it goes into G1. Once those 2 cells have divided, both of those new cells, we call them daughter cells, are going to begin to grow in the G1 phase. And then it's going to move into the S phase. Now, this phase is the point at which the DNA is replicated. Now, why is it called S phase? I couldn't tell you. Actually, I can tell you. It's the phase where DNA is synthesized. That's the word that scientists like to use for it. I kind of like that word too because I'm a keyboard player, and we call our keyboards, when we have a keyboard that generates sound and you can modify the generation of that sound to make it sound different ways, we call that synthesis, sound synthesis. So we refer to our machines as synthesizers. Maybe you've heard that term before. But the S phase is the synthesis phase, the phase during which DNA gets replicated. Okay, so you get a whole new copy. And then the cell will move into what's called the G2 phase. And this is sort of a growth phase as well, which is what the G kind of stands for, I believe. I believe I'm correct on that. I could be wrong. But it helps me remember what's going on. So I use growth as what the G stands for. But the G1 phase is just cell growth. The S phase is the synthesis of DNA. The G2 phase is where the cell then now begins to prepare for mitosis. And we're going to dig in a little deeper in this as we go through this chapter as to what exactly has to happen for the cell to prepare for mitosis. We're not going to get into that at this particular moment, though. All right, so those are the 3 phases of interphase. Now, some students think that cells get continually smaller and smaller and smaller and smaller as they divide. But they don't, because they go through the G1 phase where the cell actually grows again. And then it divides, and they're smaller at that point, but then they grow, each of them grow again before they divide. And so it keeps getting cut in half and then growing back to full size, and cut in half and growing back to full size. So I just wanted to point that out so you're not mistaking the idea that perhaps these cells divide and get smaller and smaller and smaller as each of the daughter cells divide, then their daughter cells divide, then their daughter cells divide. Some people might think, well, they must still be continually getting smaller, but that's not the case. All right. Now, let's talk about the growth phase. Oops, went too far. Well, actually, we already did. So the growth phase, the S phase, the G2 phase, they are all part of interphase. So try not to get them confused. Okay? So 3 phases that are part of interphase. Got to keep those straight. Let's move on to the section that is not part of interphase, but it's the part of cell division, and this is the M phase. Now, the M phase is also broken up into 2 stages. Okay. Now, the M phase is usually a lot shorter than interphase, and it is the M phase that causes the cell to split into 2 new daughter cells. So the first step of M phase is mitosis. Now, this is the point at which the cell's nucleus actually divides. So mitosis can be divided into 4 phases. So we kind of keep getting sub-phases in each of these phases. But mitosis we break up into 4 stages. And these 4 stages will lead up to the second part of M phase, which is cytokinesis. Now, cytokinesis deals with the cytoplasm being divided and the 2 cells being formed. So cyto refers to the cytoplasm, and the kinesis is the separation of the cytoplasm. Okay, so cytokinesis is the separation of the cytoplasm. The result is two daughter cells that each have a nucleus with a full copy of the chromosomes. Now, multicellular organisms, they actually grow by adding more cells rather than by allowing individual cells to just keep growing infinitely larger. So rather than your cells getting bigger as you grow older and as you get taller and as you get a little bit wider, um, uh, it's not a result of your cells getting bigger. It's a result of your cells dividing, going through this process. And now you have more cells than you began with. So if at some point in your life, which at some point in your life you were only about 2.5 feet tall, you had a certain number of cells that made you, that you were made up of. But by the time you grew to be about 5 foot, where you doubled in size, the amount of cells that your body contained also probably roughly doubled. Probably not exactly, but somewhere around there. So we've talked about issues where large cells have a lot of problems, right? They have problems with moving materials that they've created. So the ribosomes build proteins, and then the proteins get modified in the Golgi body and get released out of the cell. Well, that becomes more difficult as the um, cell gets larger. Also, getting materials such as nutrients and so forth into the cell, into the areas where it needs to be. If the cell is too large, it could take way too long for those materials to travel the distance they would have to travel in order to reach the locations they're needed. So cell division helps alleviate that problem by not allowing our cells to grow too big. All right, so let's get into the phases. Now, so we talked about mitosis having 4 phases. The first of those 4 phases is called prophase. And I'm going to give you a way in one of our lessons on how to remember these phases. And it'll be very helpful to you. It's called PMAT, but we'll get there. But prophase is the first stage of mitosis. Now, if you're taking notes, you might want to do this in a table. Maybe structure your table so that you have 2 columns and 4 rows. And then you should list one phase of mitosis in each row. So you would put prophase and then metaphase and then the other phases as we go. And then in the second phase, you can just kind of describe what's happening in prophase. So in prophase, actually a lot is happening in prophase. The nucleus begins to condense, and the nuclear membrane actually starts to break down. And this allows the chromosomes to become visible. Now, something else begins to happen as well, in that these spindle fibers begin to form, and they form at the locations of what we call the centrioles. Okay? So we've got the chromosomes condensing, we've got the nuclear envelope breaking down, we've got the centrioles starting to protrude their spindle fibers, and that's all going to lead us into the second phase. Now, keep in mind that all of the chromosomes had already been duplicated or synthesized during the S phase. So there's already 2 copies of the chromosomes inside of the nucleus, and that nuclear membrane is starting to break down. Okay. Now, the breakdown of the nuclear envelope begins kind of at the end of prophase, and that moves us into the next stage, which is metaphase. And during metaphase, those spindle fibers that were coming out of these centrioles attach to the centromeres, the center part of each of the chromosomes. remember that there's two copies of each chromosome. So this is one chromosome, and this is a copy of that chromosome. And the spindle fibers are attaching to this chromosome at the centromere, and this spindle fiber is attaching to this chromosome at this centromere. And we're going to find that they actually are going to get pulled apart. So again, we've got the chromatid, and we call, so this is one chromatid, and this would be another chromatid, the one that's not circled. And so we call those sister chromatids. We got the centrioles putting out the spindle fibers. We got the centromere at the center of the chromosome, and those spindle fibers, again, are going to attach. Now, not only are they attaching, but they're also lining these chromosomes up throughout the middle of the cell. Okay, so they're kind of getting relatively evenly spaced so that it makes it easier for these spindle fibers to attach to the centromeres. Okay. Now, each chromosome, again, this chromosome here and this chromosome here contains the identical information. All right, now that leads us to anaphase. And anaphase is the process where those chromosomes, the sister chromatids, are actually separated from one another. So now they're just chromosomes. They're no longer sister chromatids because they aren't paired up any longer. Okay, and those chromosomes are then pulled to opposite ends of the cell by those spindle fibers. Okay, so now we have the individual chromosomes. So anaphase is just basically the separation of the sister chromatids so that the chromosomes are then pulled to opposite ends of the cell. And that brings us to stage 4, and that is telophase. And this is where the cell actually begins to divide into daughter cells. So some of the important things that are happening here is that the spindle fibers release the chromosomes, and a new nuclear envelope begins to form around each set of identical chromosomes. Okay, so we have that here. You can see the dashed line representing the newly forming nuclear membrane. Okay, now if you recall, the M phase consisted of mitosis, which is broken into the 4 phases: prophase, metaphase, anaphase, and telophase. PMAT, P for prophase, M for metaphase, A for anaphase, T for telophase. That's how you can remember them. And you can remember their order because they do have to stay in order. So PMAT is an acronym you can use to help you remember the order of the 4 phases of mitosis. Once mitosis has completed, once the nuclear envelope has reformed around each set, a new nuclear envelope forms around each set of chromosomes, we move into cytokinesis, phase 2 of the M phase. Now, cytokinesis is the final step where the cell membrane pinches in at the center and forms the 2 daughter cells. All right, so let's look at this. We've looked at each of the stages of mitosis separately. Let's look at them kind of together in somewhat of an overview in order to get a sense of the overall cycle. Now, it's important to make note or make some mental images or take a snip or something of these images, because you will see these images on homework and so forth. What you're seeing here is an artist illustration of what each phase looks like. So interphase, the G1 phase, S phase, and G2 phase, all part of interphase. The chromosomes are they look like they're in a jumbled mess. They're really not, but it kind of looks like a plate of spaghetti if you were to zoom in. Let's go ahead and do that. Okay, so it looks a little bit like jelly, and this is what a cell typically looks like. This is actually a picture of a cell that's in S phase, or in interphase, sorry. Okay, so that's G1, S phase, and G2. And then let's move on to the first stage of mitosis. Oh, hang on a second. Sorry. I got to undo the. There we go. All right. So the first stage of mitosis is prophase. And here's what it looks like. Okay. So here you can see that we have the chromosomes compacted into sister chromatids. And we've got spindle fibers beginning to form. And this is what it actually really looks like inside of a cell. Okay. So it looks a little more organized. If you compare it to interphase, it does look like maybe it's a little more organized. So this is prophase, and this is what it looks like. Okay, and then we look at metaphase. And metaphase looks even more organized than prophase, as you can see here. Prophase, everything's still kind of jumbled in the middle, but in metaphase, all of the chromosomes are now kind of lined up. So they form more of a line here, a row. Okay, instead of being like a bunch, all in a bunch, they're kind of lined up. And that's what the illustration that we looked at earlier also showed. So that's what metaphase looks like in an actual cell. Anaphase, we begin to see the chromosomes actually being pulled apart. So it goes from metaphase to anaphase, where those chromosomes are being pulled to opposite ends of the cell. Okay. Now there's no nucleus, so don't get the impression that this outline here is the nucleus itself. That's actually the cell. So you can actually see more than one cell in here. There's a cell here, a cell here, a cell over behind the illustration, probably another one right here. This is just one cell. Okay, so the chromosomes are being pulled apart, the chromatids rather, are being pulled apart to opposite ends of the cell. Which brings us to telophase. And let's have a zoom in here on telophase. And as you can see, now the nuclear envelope is beginning to form, and it's beginning to pinch in just a little bit. Can't really see the nuclear envelope forming, but you can see that the cell's beginning to get that 8 shape. It looks kind of like an 8 to me, anyway. Okay. And that's going to bring us to cytokinesis, where we now see the cell actually separating. Okay, and that's cytokinesis. And that is an overview of the cell cycle.